listeners, and welcome to the DrGPCR podcast. Did you hear about the DrGPCR University? It's the ultimate learning platform within the DrGPCR ecosystem. Here, you can expand your knowledge, connect with experts, and stay informed about the ever-evolving world of GPCR research. We offer both live and on-demand courses covering everything from GPCR pharmacology data analysis to drug discovery strategies. Our courses, like those that focus on using Graphite Prism, help you master the pharmacology data analysis framework, navigate the complexities of GPCR research, and improve your work's quality, reliability, and efficiency. Join us and elevate your GPCR expertise today. And now, let's dive into this episode.
Hello, everyone. This is Yamina from DrGPCR. And today I'm very excited because we've been trying to schedule this podcast recording for quite some time now. I have with me Dr. Mikel Garcia-Markos. Mikel, thank you so much for joining me today.
Yeah, thank you. No, I'm happy to be here, and I know that it's always difficult to arrange these things. I'm happy to finally make it, and let's see if I can say anything useful.
I think that's not going to be a problem. So why don't we start at the beginning? Before we hit record, I was just mentioning that we both live in the Boston area. We haven't had a chance to meet up. So tell us a little bit more about who you are and what do you do, and then we can go from there.
Yeah, so I'm a professor in the Department of Biochemistry and Cell Biology and the Department of Biology at Boston University. And my lab in general works on transmembrane signaling through G proteins, the G proteins that are activated by GPCRs. So I do some work with GPCRs, but I'm more G protein centric. And I don't know, speaking of background, how did I end up here? So from a career perspective, so my lab has been active for about 12, 13 years now. I ended up here after doing a postdoc at University of California, San Diego with Marilyn Farquhar. And before that, I'm an immigrant. So I came to the US for my postdoc. And before that, I did my training between Spain and Belgium, which is where I got my PhD.
Interesting. And your training was in what discipline? Was it biology?
Yeah, it was also in general signal transduction, receptor-mediated signal transduction. I did work on purinergic receptors, but not necessarily. I mean, purinergic receptors are divided into the ones that are metabotropic, which activate G proteins, and the ones that are ionotropic, which are ion channels. So I work on both of them. So I was not very focused on GPCRs. Which is funny because when I started my postdoc mentor thought that I was a GPCR person 100%. So I had to assume that role when I did my postdoc.
I love it. And so you mentioned, doing your PhD and doing undergrad in Belgium and Spain. What brought you to the US? Obviously, I also came to the US for my postdoc. I think there's a lot of listeners, a lot of people who, it's anyway a good thing to move countries, but I think the US is a very popular country to come and do a postdoc. But at the time, did you specifically focus on coming to the US, or what's the story behind picking that postdoc particularly?
I had a preference for the US. I mean, my perception at the time, and probably sounds bad to say it now that I'm in the US and I have a lab in the US, the quality of research was better. Overall, and it's also a mix between higher average quality and larger number of opportunities. So that was, the driving factor.
Yeah, absolutely. And UCSD in California, not a bad place to do a postdoc either.
Yeah.
I visited once and very much loved it. Loved it there. Plus, you have a really great community there. And then once you've completed your postdoc, did you always know that you wanted to be an academic, or did you explore industry opportunities, or it was just kind of a progression of things?
So I was interested in academia, but I was also, and this is something that I tell people in training now doing postdocs is that I never got very obsessed about it. I knew that it was difficult, that I may make it or not, and either way was going to be fine. And that's, I mean, if I could give any advice to people, I think people stress out a lot about this, having to decide what you want to do. My point of view during my postdoc was I'm going to be as productive as possible. And productive means do good science, get the skills, and get published. Have something that you can show. I will try to get a position in academia, and if it doesn't work, industry is also a good life. So it's a matter of I had a preference, but I was not fixated into if it's not academia, I'm going to die. So I think if I could have gotten a faculty position, I could have also been fine.
That's why I was asking the question, because at the time, so I completed my postdoc over, wait a minute, let me think about it, because it's been a while. So I finished my first postdoc at Rockefeller with Tom Sackmar in 2016. And back in the day, I didn't know what I wanted to do. And it was this kind of a progression of things. So during my PhD, back in the day, you didn't have so many resources. So you were told by your PI and the surroundings, oh, you have to do a postdoc in order to be a professor. That's the only way you can go. So entering my first postdoc, in my mind, it was the only thing. And I didn't get a chance to even think about or look around and say, oh, if this doesn't work, then here are the other options. And that's why I wanted to bring this up, because I'm sure that whoever is listening to this podcast when we release it, there's a lot of people in the same situation. So I like your approach. This is what I want, but if it doesn't work out, I'll be fine. It's not the end of the world.
Yeah, and also speaking of, I mean, there is a lot of talk these days about postdocs and how useful they are. And I don't think it's all, I mean, doing a postdoc has value. Even if you are not going to become a faculty, a PI. Yeah. I think when you finish your PhD, to put it in some way, you have made a number of mistakes and you have learned from those ones. And doing a postdoc is your opportunity to start fresh and clean. And you can take over a project and be more independent and work on that. I don't think it's just, if you are not going to become a PI, don't do a postdoc of 5 years. But you can, a postdoctoral experience, I think has value in itself, regardless of whether that your career path is going to be academia or not.
I agree. And I can definitely say I have no regrets. I really enjoyed the experience. It allowed me to get to have ownership of the project and to create a project. And I think that independence, you may not get to it during your PhD because you're still learning.
Yeah.
So
yeah, absolutely. Yeah. Ownership is the right word.
Fantastic. So you mentioned having started working during your PhD on purinergic receptors. When did you start working on GPCRs or GPCR related proteins? Because and to your point, when you introduced yourself, you said, oh, you're interested in G proteins that are activated by GPCRs. And one of my questions in the series is, what's your favorite GPCR? But it can be anything. Just, we had to pick a name for the podcast and for the community.
No,
but if you're
so it was during my postdoc, I became more focused on G protein signaling because that was a topic in the lab. When I joined my postdoc lab, Marilyn Farquhar's lab, she had been working for about 10 years on G protein regulators. So she was one of the first people to identify RGS proteins, regulators of G protein signaling. They had done a number of screens and identified a number of potential G protein regulators, and I started working on one of them. So that was when I became more focused on G protein regulation. And that's what has become, my lab has become that. I mean, we have a general interest in knowing what are all these things in addition to GPCRs that control and shape G protein signaling pathways. The question about your favorite GPCRs, so I have I guess people will probably answer with the one that they are working on or they have worked on or something like that, because I have not been very GPCR centric. I tend to think about GPCRs that other people work with and that they are weird and they are different. And recently, I mean, this came up in another context, and I said that as right now, the one that I think is really cool is GPR 158, which is, I find it very interesting. I mean, it's this in part because I have seen Kirill Martemianov work on it for many years, from starting from not knowing anything about that GPCR to knowing a lot about it. From, I mean, it was not known what the ligand was, what was the function, how, whether it regulated G proteins or how, and now all those things are falling into place, and it's quite amazing. I mean, it's a metabotropic receptor for glycine. It forms a dimer. It activates G proteins indirectly through an RGS gap. So it does all the crazy things. I mean, that's things that you would have imagined, let's say 10 years ago. And that's why I find it right now it's kind of a very cool and weird GPCR.
That's the hot GPCR. I love it. I think it's really great to have the opportunity to kind of dive into a specific GPCR and be able to ask the right questions. And I'm always fascinated. You mentioned that this particular receptor activates indirectly G proteins. I mean, who would have thought? And I feel like these receptors in general surprise us. And we know that there's this number of receptors. We've drugged a certain number of them. There's many more. There's the olfactory system as well. And there is a classical mode of activation. But when you think about all the physiological functions that the receptors have to carry through in the body, I'm not surprised when we discover that GPR 158 you mentioned?
I think that's the number.
Yeah.
I hope I'm messing it up.
That indirectly activates G proteins, which I think is fascinating. So when you were a child, I want to go back a little bit in time. Did you always know you wanted to be a scientist, or was there something else that you wanted to do?
No, I mean, it's very sad. I mean, this also came up recently now in a conference. I was in a circle like this, and there was some discussion about how did you get started with science? People had very wonderful stories, and mine really sucks. I mean, I don't know. I cannot put a finger on why I became a scientist. It happened like this. So in general, so I could put it this, there are some things that took me in the direction of doing what I'm doing now, but it happened progressively. So one thing is that I think during high school, I think I felt like the humanities, to put it in some way, I think I was, to put it in some way, not very smart in the way that, or I was not mature enough to think about the bigger questions of humanity. And things that were more scientific was something that I could grasp, something that I could kind of understand. And it also had defined answers. So something that bothered me a lot when I was a student in high school, for example, was topics in which the answer could be many different things, and then they could evaluate you however they wanted. Whereas science, in many cases, was more defined. Now that I have become a scientist, I have a completely different perception of science, and I think it's, especially biology, is subject to interpretation a lot. But back then, that's a little bit what took me in the direction of science. And then I don't know, I had a sense that I liked biology and chemistry. I did not like the part of biology that was about looking at plants and animals. I looked more, I was more attracted to molecular things. So that's why I ended up doing, in college, I started biology, and I did like a specialty last 2 years on biochemistry. And but I cannot I cannot say, I got a chemistry kit when I was 8 years old, and that was it. I read a book about the biography of scientist X. I
Yeah.
didn't have anything like that.
I love it. It's interesting because I was listening to you mentioning this, and I can relate to the fact that humanities back to that point of view. You can't grasp it, you can't calculate it, and you didn't have a definite answer. And I think it uses a different part of the brain. And I think us scientists typically want an answer, want a number, want a molecule, want something specific, something defined. Whereas humanities, poetry and literature, beautiful, stimulates the mind, but it's not as it's different. It has a different. As scientists, we have a different propensity, and absolutely can relate to that. I
Yeah,
have a
but
9-year-old.
that I was not ready, to be
Yeah,
honest.
yeah,
I
yeah,
mean,
yeah,
it's not high school Mikel is not like Mikel today. I mean, today I can think about the bigger questions about who we are and what happens to
yes,
us in life and
yes.
so on. When I was in high school, my brain didn't function that way.
Yeah, I think a lot of kids are like that, and I can remember being having the same disposition. I have a 9-year-old who absolutely has that disposition, who only wants to read science books,
Mhm.
and that's about it. Um, how, all right, so let's fast forward. You've completed your PhD, you're completed your postdoc, you are looking for faculty positions. Can you tell us a little bit more? I know it's been 10, 12, 13 years, but can you tell us a little bit more about that experience? How was it at the time? And if you can also contrast to how you see faculty positions today.
Yeah. So it's interesting. I think I did all the things that I tell people don't do, meaning that I didn't plan it well. I applied out of sight. I mean, the US, at least in the US, there's a very defined cycle. So now is the time to send applications. Now there are a lot of open faculty positions. I sent my applications like in January. So it was bad. Why? Because my son, then there's this factor about life. My son was born in October. So I couldn't go with that cycle. At the same time, I was also applying for my green card. So that was a very tough period for me. And I'm mentioning it because I think it's an example that even when things are suboptimal, they can work out. And that going again back to the point about plan your life, to what extent to plan your life and your career moves. It is good to plan things and have strategies and have defined goals, but sometimes life gets in between and you just have to adjust. In my case, it was that I had to, in terms of the process, I didn't do it the way I could recommend people to do it, but I had to do it that way. I got kind of lucky. I didn't send many applications. Today, I would recommend people to send many applications every cycle and even consider, and I did only one cycle. So that's all my advice these days is that it's even good to send applications early, one cycle early, just to test the waters and get a feeling of whether maybe they will call you to go to a Zoom interview and get a sense of how the process works, because just going through the process is something that is very useful. Even if you know that, oh, I'm not ready, I need an additional paper, or I need my big paper, or anything like that. I think that's fine, but there's another factor about, it's not about how you look on paper, but how well you do the process, because the finding a faculty position is a process. And until you experience a little bit of it, you're not really prepared. I mean, there are many resources these days that I think I didn't have 12, 13 years ago. People talk much more. There are forums. You can get a sense from other people. They can explain you how a chalk talk is. When I went into this thing, I had no clue. It was, things just happened. I was improvising every step. So that's it. So I got kind of lucky. I got to interview in a place which was this one that I liked, and they made me an offer, and I took it. So I don't know what else to say. How different, I mean, to these days, I think it's similar. I think it's equally challenging. I mean, when I got my position, there were 400 applicants for the same position. I think they made two offers. The last search that we did in the department was less than that. I think we had in the range of 300. But I give these numbers not to discourage people, but the opposite. It's normal. I mean, it is. These are numbers that many people get filtered out for different reasons. It's about the fit with the position. You cannot anticipate when you're an applicant. It's very difficult to know exactly what they are looking for, and because even though you see an ad, it's challenging to know the fit. So that's why I recommend sending many applications and getting experience with the process, because that helps a lot. Finding yourself in the situation of having to answer questions of a screening interview or going in person, and I think that has a lot of value.
I love it. I think to your point, after talking to so many people, I don't think I've met anyone who clearly planned it out from day one. It was just, I've been a postdoc for X number of years now. Now is the time. Funding is running out, and let's go for it and do it. I like the advice of trying to, submitting applications one cycle early to get a sense for the entire process. I think it's great advice. So thank you for that. Awesome.
It's case by case. I mean, some people are superstars and they just need to go for it, and they are going to get multiple offers. Other people may need, and they may have a natural ability to do interviews well. But I think the majority of people need experience.
At the end of the day, you just need one. You need one position. So when you think about it, that's why the numbers game of give. When I said hundreds of applicants, I don't want it to sound discouraging. It's probably the same. So out of those hundreds, I mean, probably there is more than 50% that are applying for all the same positions that you are sending. So there is a lot of you are competing against the similar pool continuously. And it's true, you just need one. And that one can happen because of reasons that you cannot imagine, which have to do with what that institution is looking for. Yeah. The other well-made point was about not knowing what they're exactly looking for. Although there is a job description or a description of the position, it's very hard to articulate specifically what people need. And it's typically, job descriptions in general are a wish list. And you have to meet people, you have to be there, you have to shake somebody's hand to kind of get a sense, and the talk and all of, and even the dossier that you would submit with the projects and information about yourself. It really depends. I think it's the same thing whether you're in academia or industry. It's all about being able to come through to that person, but also connecting with that person. I always tell people, if you're looking for a job and you're interviewing the company just as they're interviewing you, and it's about finding that match. And I'm sure it's the same thing in academia as well when you're looking for a job.
Yeah, and the other point that to mention about search is that there is, I don't know how the perception is from the outset that these searches are very, they have defined criteria and so on. These committees, they're made by faculty, and each one of us, I mean, we are weird in general, and we have a lot of things in our heads, and the search can be about X, but there can be voices in that room, in that committee, that have nothing to do with what is advertised. And there are many things that can happen during discussions and conversations that are completely unpredictable, but completely unpredictable for the people in the committee, not even for the candidate. So it is something, when you look for faculty positions, you have some control over your part. So that's focus on that. Don't think too much about what's going to happen on the other side, because what happens on the other side is chaos.
Yeah. No, that's true. You can only control your own 3 feet, your own reactions, your own output scientifically. So great advice. Since we're talking about advice, any additional advice to scientists who want to be working on GPCRs, G proteins, anything that you have learned in the past 12, 13 years being API?
In general, I could say that the field is a good community. So general advice is interact with people. In general, they're going to be good people. That has been my experience. So I came, my lab, I started working on G proteins during my postdoc, and the lab worked on G proteins, but it was not. I think my PI didn't feel particularly connected to the field, but I realized that, by going to conferences and meetings and just talking about your work, is people are open to listen to you and give you advice. And this like buzzword of networking actually means just go and talk to people. So it's something that happens easily in the general field of GPCRs and G proteins. I think it's a kind of friendly and collegial in general. So my general recommendation is that go out and talk to people. You're going to gain things.
Agreed. I think we're in a fantastic field. People are typically nice, and it's okay to send out an email. It's okay to say, hey, I'm going to this conference. I saw you're speaking. Would you have 5 minutes to have a conversation? And I think that also is helpful when it comes to finding your next faculty position, because then you've developed relationships with other scientists who may be going through the same process as you are, or somebody who can say, oh, actually, we're looking for someone in the department. Let's have a conversation.
Yeah. No, I think it happens naturally and it's quite organic.
Yes, agreed. One other thing I wanted to ask you, so when you started out your lab, and you came in with fresh ideas on working on G proteins, how did you think about setting up your lab? How many projects did you start with? How many projects do you have now? What's the dynamic and the philosophy in the team?
I guess it's kind of chaos.
An organized chaos.
Yeah, I mean, you control us. How you start trying to, I mean, the first thing that happens when I started, and I think it happens to a lot of people, is that you are driving a car at 100 miles per hour at the end of your postdoc, and you start your own lab and you stop. It's like hitting a wall, because everything is the machinery is functioning while you're finishing your postdoc. You are probably the most functional and efficient scientist that you know. It's you have full control over your projects. The lab is a functioning lab. All the details about having all the reagents, having all the techniques, having all the advice, all of that is in place. You start your own lab, and I remember, I mean, mini preps were not working, and it's that feeling that you were going really fast and suddenly you stop and have to restart, because the small things will slow you down. So that's from a practical perspective. That's the first feeling, is that you were achieving so much on your own that when you start to build your team and you feel like things were going even faster when I was a postdoc, it's something that you have to realize that it's normal and that it happens, and it takes time to create some scientific critical mass in the lab, that things start to take off. And in terms of projects and so on, so the beginning was a natural continuation of my postdoc, but something that you realize very early on is that science takes you to places. So you have this preconceived idea that the project is this one, and you start getting data. Two things influence what happens: the data you start to get and the people who get in your lab. Because very early on, they will come. I mean, I remember some of the first postdocs I had in the lab came with somewhat different background, and that took us in a different direction. It just happens. I don't know. It's very difficult to explain. So to this day, I still try to keep like a cohesive general theme, but things evolve on their own.
Yeah. And I think, you touch on an important point about having people joining and from different backgrounds and adding to it, or taking the project in a different direction. And I think that's the beauty of science. Working on GPCRs or any membrane proteins or any project is challenging. It's not one person, it's a team effort, and having a multidisciplinary approach by bringing in people from different fields, whether they're people who are working in the lab or who you collaborate with, just allows you to get a different perspective and develop different methods or better methods, or ask questions that all of us kind of had at the back of our heads, but never really stopped to actually ask those questions. So I love that. I really love that approach.
Yes, that is in terms of bigger picture, bigger points, but even a small thing, someone can come and do something that you are doing is slightly different, but it's much better. And that becomes an integral part of your lab. And when you look back and you think, I'm glad we made that change 10 years later, because now you think about all the things that you could have not done if that change didn't happen.
Yeah.
So,
yeah. And to your point, when you're a postdoc, you typically work in a well-established lab where everything is there for you to do your work. And when you basically set up your own shop, you have nothing. And I was laughing when you mentioned, mini preps didn't work. I do remember when I joined, when I first started working in the lab, it was a summer internship. So initially, I worked on chemokine receptors, and we were trying to make mutations in the drive motif, one amino acid at a time. And this was quite some time ago. And so we were using this Kunkel method of mutagenesis, where you make a single strand of DNA, order oligos, and we didn't have ATP in the lab. And so we actually went and looked into some kind of a restriction enzyme buffer that had ATP, and used some of that in order to start the reaction. Needless to say, I made, I think, 6 mutants. It took me 2 months to make 6 poor mutants, and didn't even get a chance to characterize them properly, because by the time I had the mutations into CXCR4 chemokine receptor, and we expressed and we had enough DNA, the summer was over. So I ended up continuing to work during the school year, but also did a master's to characterize these mutants. And I do remember interviewing for this position for the summer internship, and there was nothing in the lab. There was the PI, a laptop, a phone, and bills everywhere. And, and it can be challenging, but also in a good way.
Yeah. Yeah, no, the other thing about starting your own lab is how many hats you have to wear, things that you didn't do before. You will still be doing bench work. I don't think it's very difficult to make a successful lab if you are not the person still doing a good chunk of the experiments initially and training people directly. The very first people who come to your lab, I think they should be trained by yourself, very hands-on. But on top of that is all the bureaucracy of safety protocols and ordering, and each institution is learning about how an institution functions. I mean, there are a million things that I don't know about my institution today, but when I started, I didn't know anything. I remember, how do I get internet? It was one of my first questions. So you face all those things and are new, and you also need to be thinking about money and how to make money and managing people. I don't think I didn't get any formal training on managing people, and that has been really the most difficult thing, is how to lead, how to be a team leader. It has taken, for me, first assuming the role of a leader was something difficult. When you start your lab, you still feel like you're a postdoc. I mean, I was just thinking, I'm this guy who is doing experiments and is talking about science with other people in this room. And it took me years to realize, but I'm supposed to be something else. I'm supposed to be, telling people what to do, how to do it, and have a different role, a role of leadership.
Yeah. Yeah. And it's a difficult thing to do, because at the time where you start off, whether you're starting your lab or you're starting a team in general, you're the expert. And anything that anyone joins in, anything this person has to do, you know how to do in minutes, and it's going to take them hours.
Yeah.
To give an example. And it's very hard to let these things go and say, no, my role now is to lead. My role now is to teach. And so I'm going to tell the person what to do, let them make the mistakes, and give them the opportunity to grow into that role. And I think this transition, as you mentioned, is very difficult, but it has to happen, because in my mind, that's the best way to provide the next generation the knowledge and the opportunity to grow into leaders later on.
Yeah, but the conflict there, the internal conflict, and this is for anyone who is thinking about, setting up their own lab, is you will think about this theoretically, that this is the idea. I'm going to lead, and I'm going to teach, and what matters is what is going to happen after, But at the same time, you're under a lot of pressure, because you need to get things to work, and without, producing, you're not going to be able to get funded, and you're not going to keep going to exist as a lab. So there's a lot of internal conflict with all that, and it's a lot of psychological pressure, and that's something that I don't know if people talk a lot about it. It has been, that's another thing that is a challenge, and it takes a lot of your energy.
Yeah. I don't think people talk about it a lot. So how about we talk about it a little bit? Any lessons learned when you got to that point where you said, no, I'm not a postdoc anymore. I can't be in the lab. And anything that you wish you could have done differently?
I think it worked out okay. I didn't plan. I didn't plan it. So I think it happened. I realized that. So when I was more in the lab, was it bad? No, not at all. I think I was very useful at that time. It created a lot of stress situations for me and other people. I think so, because the pressure was higher for them to have me seeing things and giving advice in real time. And for me, because I saw too much. And that's something that you also, at some point, it's a good idea to just separate yourself from the source of, let's say, stress. But when that's supposed to happen, it's what no one will be, I don't think anyone can know. I mean, I got to a point in which I decided I have to change the style of how I'm doing things. I have to progressively go and spend more time in my office and doing the things that are more useful for my lab, which is I'm better than any other in writing things, or things that other people cannot do, and just give up doing experiments. Even though I love to do experiments, I think it's wonderful, and I always say that I didn't know it, but my time as a postdoc was probably one of the best times of my life, in the sense of I could do science and only think about experiments and what they meant, and think about the next question, without having to spend a lot of time on doing this fragmented life, in which I think about the science and bureaucracy and applying for funding and all these things at the same time. So it's very difficult to know in your progression, when you have your own lab, when to make that change. I think it's useful to be in the lab in the beginning, but at some point you need to start giving it up and let other people do the experimental work, and so you can focus on the other things that you're the only one who can do.
Yeah, and also, it's for the benefit of everyone, and by that I mean for the lab to make sure that people have a job, have the opportunity to train with you, and also move science forward through your team.
Yeah.
I do remember, I have to say this, I hope he won't hate me for this, but I do remember when I started in Nicolaus Iveker's lab at the beginning, he spent a lot of time. I mean, I had a lot of I was his first student ever. And for a summer intern, for his PhD, for his master, everything. So I had the opportunity to learn a lot from a scientist who had really great bench, discipline, and also really great hands. But I do remember once he also went through this transition of hiring more people and spending more time on focusing on what he was in control of and making sure that we had everything that we needed. And when he would do sometimes experiments, and I do remember one time he ran a PCR, and he was wondering what happened. Well, he realized he didn't close the Eppendorf tubes. And we said to him, Nicholas, this is this is a sign that are no longer needed in the lab, because you're really good at what you're doing, and you have to give that up. And the minute also when he realized that he didn't know where things were in the drawers in the lab, that was the cue.
Yeah.
Of saying, okay, I need to give this up.
Yeah, no, I have similar anecdotes. I remember telling someone, I will set up your Western blot transfer, and I set it up the opposite way, so so I have done all those things that you mentioned. I'm capable of doing all the PI things.
I love it.
Yeah.
Anything anything to add from a leadership perspective? You're right. I don't think that during our training in general, no matter where you end up in academia or industry, we are trained to manage, to work with other people, to be responsible for other people, and to lead. It's not about management, it's about leadership. What are maybe the top 3 things that you've learned over the past decade or so from a leadership perspective?
That I don't know much about it. That is something that you need to keep working on constantly, and you should be exposed to it as early as possible. So my advice is, any opportunity that you have to mentor someone in the lab, take it, and think about it as not that my PI is giving me this extra task that I have to look at this undergrad, and just take it as something, as an experience that is going to be useful. You're going to start learning how your first experience, you will think you're learning something, but you're just learning how that person you're supervising is or behaves. And that's your first experience. And you're going to need many of those to start getting a sense of people and how people are different, and how what works with one person doesn't work with the next one, and things like that. And then I have started specifically reading about management. It's not so difficult to grab a book about management in industry, even if it has nothing to do with science, and quickly you realize how things relate. So it's all the same. It's a different context. It's not, I read books even about how, I read one very interesting one about how, which is very relevant for science, I think. It's about management of teams that are multicultural, that include people from different nationalities. And I think that book was eye-opening to me, because I realized that a lot of things that I was assigning to personalities in my lab, they actually had a very clear root in cultural aspects of how people have been raised in different countries. And that's the nature of scientific teams. I don't think I have 2 people with the same nationality in my lab.
Yeah. Yeah, no, and you're right. We just went through, as a Dr. GPCR team, through 2 sessions of a team building activity. And the first thing we did is we brought in someone to help facilitate the discussion. And everyone was able to take a personality test. We did the MBTI. Everybody got their reports, which were not shared across, but we were able to actually get a sense as to how people are and who likes to work how. And we got a sense of what is the profile overall of our team. And it turned out to be imbalanced in the sense that we have more introverts than extroverts. We have a lot of people who are precise, like to what they call judging, in the sense, not judging others, but in the sense that they're very punctual and they're very specific. So, for example, I'm going to give the example of my profile. I turned out to be extroverted only at 60%, so I can balance between the two. But there was a saying for my category that our instructor gave us, and it went something like, if everybody would do what they're supposed to do, the world would be a better place. And when I heard this sentence, I said, yes, this resonates with me, but it's not the case for everybody else. So the goal with the 2 times 2 hours we sat down together is to learn to consider others and to learn to flex. If you're an extrovert, you can understand how an introvert might think or might work or might react. If you're an introvert, you can come back and say, okay, I am this way, but this person is that way. Therefore, I need to adapt to that.
Yeah.
And I think that helps a lot.
Yeah, any in general, any activity, anything that allows you to put yourself in someone else's situation, approach that, I think is very helpful from gaining leadership skills. Science is something that you become so specialized that it's the extreme of focus on a particular topic. And dealing with managing people, it's almost the opposite. It's about being able to just get out of yourself and try to put yourself in the situation of 5, 6, 7, whatever other many people you have in the lab, and they are very, very different from you. They have different age. They're from different generations. They are from different countries. They made different personalities. So it's all very complicated.
Yeah. Like science isn't. I think it can be just as complicated as science, and it's all about being open-minded and giving the opportunity to everyone to bring in their expertise, to bring in the richness of their cultures, of their personality, and putting that into good use. And I think it's a difficult thing to do for a leader. But that's,
I mean, in terms of leadership, the other thing is that creating a group identity is super important, and you have some control over that. And a way to facilitate that is to kind of from the beginning make people believe and trust you that they are not on their own, that they are going to be working with other people, and try to maximize the opportunities to collaborate. In collaboration, I think it has 2 sides. It has the side of productivity. I mean, when people collaborate, from my perspective, it becomes much more productive because they generate more data, just to put it simply. But for the added benefit is that as soon as 2 people, just to mention, let's think about the smallest possible group, which is 2. As soon as someone can generate data that can be presented to another person like this and be understood without an explanation, that creates a bond, and that creates much more joy related to the process of discovering something. Because going back to being highly specialized, what happens to us, to scientists, is we live in this internal universe about these very specialized topics of which we know a lot about, and we cannot talk about it to anyone. I mean, if you think about it, it's you live within. So if you have someone that you can go to and tell them, look what happened to my experiment, and you're using only those words, and the other person says, "Wow, that's cool." I mean, it's wow, that's what you have been working on for 3 months, and actually, it's working out, and this is very exciting. That makes it so much more fun, and that also contributes so much to generating that feeling of team of working with people.
I love it. No, I absolutely agree with you. I love it. And you're right. We speak a very different language as GPCR scientists to any other scientists. And you're right, if you get to a point where you just show the figure and the person understands and can appreciate it, that is a bond. That is definitely a bond. Lovely. All right, we're almost at time. We talked about advice for young scientists. We've talked about academia. We've talked about your trajectory. What I'd love to conclude on 2 things, actually. If you can tell us what are your top 3 aha moments that changed your trajectory as a scientist, that led you to where you are today?
I don't know. I mean, one would be during my postdoc, I was starting to work on this poorly characterized protein that was known to bind to G-alpha subunits, and it was not completely clear what it did. And I remember I did like a boring experiment, which was mapping what region of the protein was responsible for binding to G-alpha subunits. And at that point, I remember going back home, and I did a blast search of that region of the protein, and there was like a conserved sequence. And then I realized this sequence looks like something similar to a peptide that had been described by David Siderovsky's group. That was a synthetic sequence that could bind an activity protein. So I connected the 2 things, and I thought there is going to be something here. And meaning that this protein that we don't know what it does probably has this sequence, this motif. It's what makes it bind to G proteins and regulate G proteins. And I was excited because when you have that, it's very easy to envision the 10,000 experiments that you're going to do, because you have your protein, you have very clear hypothesis, you're going to start making mutants to disable that function and so on. So that was exciting. Actually, that became my postdoc and became, like probably my first R1 was on this topic. So it was really a moment that was before and after. And it came out of a relatively boring experiment. I can say that's the only one that I would classify as a moment, is just connecting those things that I have identified this, that I don't know. I mean, it didn't have a particular goal or use, and connecting it to something that was out there in the literature, and which drove the direction of the project.
I love it. And this is exactly what we were talking about. When you finish a PhD, you've learned what you could learn. You get into a postdoc and you take ownership of your project, and the sky's the limit at that point. All right, last question. What should we title this episode?
Oh.
I know it's a tough one. It is, I've just to give you context, I always struggle with the titles because we talk about so many different things. And
this one is almost all but GPCRs.
Yes, we could call it. I love it.
Because I don't think we have that much conversation with GPCRs themselves. Maybe that's
all but GPCRs. I love it. I think we should call it that. And then maybe we should have another recording at some point to actually talk about GPCRs. I would love to have you on as a guest. We're gonna have more scientific programs coming up in the next year, so I will be definitely reaching out to you and have you on for that. But I'll send you an email. We can have a actual GPCR conversation. This was lovely.
Yeah, thank you so much.
I need a favor. If you enjoyed this episode, please click the like button. And if you like the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribers yet. One click goes a long way. All the information about DrGPCR is in the description. Find all our fantastic features and everything you can benefit from becoming a premium member. And while you're on it, check the DrGPCR University courses and share them with your network, colleagues, and friends. We also want to hear from you. Is there anything missing from our ecosystem? Can we make it better? Email yamina@drgpcr.org or email our team at hello@drgpcr.org. Thank you so much for joining us in this podcast episode. If you're still there, we also want to take a minute to thank our team members: Yamina, Attila, Montserrat, Cam, John, Yatsu, Mark, Ivana, and Andreina. If you like our podcast, you can leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials and subscribe today to our monthly newsletter at ecosystem.drgpcr.com/newsletter-sign-up. Until next time, and stay safe.